Today class we are going to discuss about the dinoflagellates. See, these are the second type within protestant organisms. Right? See, these are also grown in marine, means which are mostly grown in the oceans. Right? See, the very first point here, mostly grown in the marine water. Right? And dinoflagellates, din means two. Remember, these organisms having two flagella and the body divided into two parts. The organism body divided into two parts. See, the organism also having two flagella. These are also called dinoflagellates. Dinoflagellates. See, the body divided into two parts and organism having the two flagella. Right? So these are called as dinoflagellates. And remember, I drawn here some of the plate-like structures, right? Here the blue color structures are there. The plate-like structures are nothing but stiff cellulose plates. Stiff cellulose plates. So, we know that dinoflagellate cell wall is made up of with stiff cellulose plates, means which are having cell wall, right? See, the cell wall is made up of with the stiff cellulose plates, and the organisms are having the flagella. These are having flagella, two flagella, right? And body divided into two parts. Between these two, there is a gap is there. Between these two, there is a gap is there. That gap is called as, means body divided into two parts, right? Between these two parts, one gap is there. That gap is called as furrow. The gap is called as furrow, right? And so how these two parts of the body connected? Means I said, the total body divided into two parts. Sir, how the parts are connected here? How the parts are connected here? See, there is a muscle portion is there. That muscle portion is supporting for the connection of the two body structures. Right? And come to the next one, flagellus. Remember, these are eukaryotes. Flagellus. These are eukaryotic flagellus. Right? Because these are eukaryotic cells. And the prokaryotic flagella is different from eukaryotic flagella. Here there is a 9 plus 2 arrangement is there, right? And here transverse flagella is there, longitudinal flagella is there. See, this is one of that transverse position, and this is a longitudinal position. The flagella attached to the body, one is at the transverse to the body, transverse position. Other one is the longitudinal position, right? So, based on the position, transverse flagella is there and longitudinal flagella is there. Right? So, this is the structure of the dinoflagellates, and these are photoautotrophs which are having the chloroplast, not chloroplast, chlorophyll vein, and which have the capacity to perform the photosynthesis process. Which have the capacity to perform the photosynthesis process. Right? And we are already discussed about this point that the cell wall is made up of with the stiff cellulose plates. Right? And other portion, movement of these organisms. The movement is called as spinning movement. The movement is called as spinning movement. All of you remember, this is the bacteria. The bacteria. Okay. This is the bacteria. The bacteria having flagella. So the bacteria moving towards this direction. Right? This is straight movement. But within dinoflagellates, movement is not the straight, because here flagella one is at longitudinal position, other one is at transverse position. See, the movement is quite different. For suppose if the organism want to move from here to here, from here to here, the dinoflagellates want to move from here to here. Here the movement is like this. So this kind of movement is called as spinning movement. These are called as whirling whips. Somewhat the motility of these organisms are quite different from remaining all. That's the reason why these are called as whirling whips. These organisms are called as whirling whips. That is about the movement of the organisms. And coming to the next one, colors. We already discussed these are the very beautiful organisms because which exhibit the different types of colors. Here the colors are red color is there, blue color is there, green color is there, yellow color is there, pink, brown. So different types of colors are exhibited. See, the pigments are different, the colors are different. Whatever the organisms having the pigments are there, the pigments are different and the colors are different. Right? And the very important point in this concept is the mesokaryon. We know meso. Okay, first of all we know karyon. Karyon means nucleus. Karyon means nucleus. We know U means true. U means true. Here meso means false. Meso means false. So this is false nucleus. Whatever the dinoflagellates contains nucleus is there, that nucleus is considered as false nucleus. So why this is false nucleus? Remember, this is the nucleus of the dinoflagellates. Always the DNA existing. Always the DNA existing in condensed state. The DNA always existing in condensed state. See, always DNA present in condensed state without need of stimulants. Without need of stimulants. I'll explain. Okay, just this is about mesokaryon and this is a normal cell. And this is a normal cell nucleus and this is a genetic material. During the cell division process, this is a chromatic structure we know. This is a chromatic structure. And during the cell division process, the chromatin is interacting with that histone proteins. Many number of histone proteins are there. Histone proteins are here. There is histone proteins. There is an H1, H2B, H2B, H3 and H4. These are the types of histone proteins. And chromatin and histone proteins, both these are combined. Chromatin and histone proteins are combined, which forms the chromosomes. So this is a very general concept within the normal cells. There is a chromatin and histone proteins, both these are combined, produces the chromosomes. See, the interesting point within the dinoflagellates, okay, mesokaryon is formed within the dinoflagellates. See, this nucleus we consider as true nucleus because histone proteins condensation and decondensation. During cell division process, separation of chromosomes. So there is an event certain. But within the mesokaryon, this is the true nucleus. Right? Within the mesokaryon, this is the false nucleus. Why this is false nucleus? Because whatever the genetic material is there, the genetic material is always existing in condensed state without any need of histone proteins. See, other than histone proteins, some of the non-histone proteins also there, which are also supporting, but here histone proteins are absent. Then the genetic material is always existing in condensed state. During cell division also existing here condensed state. That's the reason why this nucleus is called as false nucleus. This nucleus is called as false nucleus. Right? And some other points also there, means bioluminescence of dinoflagellates and the remaining properties of the dinoflagellates we will discuss. Yeah, coming to the next one is bioluminescence. Bioluminescence is nothing but some of the living creatures have the capacity to emit the light. Some of the living organisms have the capacity to emit the light. So the process is called as luminescence. Okay, the luminescence is done by emission of light is nothing but luminescence. The process is done by the living organisms. That's the reason why this one is called as bioluminescence. See, we well-known example is the fireflies. Fireflies are the best examples. Fireflies are the best examples for the bioluminescence. But the fireflies are not belongs to the dinoflagellates, but The mechanism is same. The mechanism is same. See, within the fireplace or any organism means here maybe the dinoflagellates. So first of all, here I will explain based on the fireplace. See, there is a chemical energy there. The chemical energy is converted into light energy. When we see the fireplace at the night time, okay, there is a light on and light off. Light on and light off mechanism will be takes place. That is nothing but when the energy is available inside the cell, the energy means chemical energy is converted to light energy. The chemical energy is converted to light energy. So how the energy conversion takes place? How the energy conversion takes place? Remember, there is a luciferase enzyme. There is a luciferase enzyme is there. That enzyme and presence of luciferin substance. So presence of luciferin substance and luciferase enzyme utilizes the ATP. ATP hydrolysis takes place. Then produces the light. See, here chemical energy is nothing but ATP. Chemical energy is nothing but the ATP. So presence of ATP, the luciferase enzyme and the luciferin substance, the chemical energy is converted to light energy. Okay? The same enzymes and the same mechanism, the same components, which are also present in the dinoflagellates, and these examples are very important. Nactyleucum, Gonolix, Pyrodinium, right? See, these are the organisms. These organisms also have a capacity to exhibit the light. See, I said so many times when we watch the National Geographic channel, some of the shows which are related to the dinoflagellates. When we watch dinoflagellates related shows, okay, during the night time, different organisms exhibit their different colors of lights. These are growing within the ocean, okay, especially which are grown in very dark regions at night time, right? Very dark region, which emits the different colors. What we discussed, whatever the red, pink, right, and there is a brown color is there. Different types of colors are exhibited by these organisms. See, all these organisms also emits the light. So presence of that pigments and the type of color is emitted. For suppose the organism naturally having a red color, presence of the light, very glorious red light will be produced. Right? So that is about the chemical substances and light, and these are the organisms which produces the light. So what is the use? What is the use to the organisms or to the dinoflagellates by the bioluminescence process? Remember, one is food hunting, supporting for the food hunting. See, these organisms, food hunting is supported by the bioluminescence and escape from predators. See, sometimes when these organisms are emits the light, whatever the predators are there, the predators are afraid. The predators are afraid. And the predators are away from these organisms because suddenly these are emits the light. Then they are afraid about the light. Some organisms are afraid about the light. So that is about that escaping mechanism from the predator, right? That is about the bioluminescence process. And see, coming to the next one is the red tides in Mediterranean Sea. We discussed already here the Gonolix, that is the organism. Gonolix organism involved in the rapid cell division. The cell division done by the binary fission. Binary fission means we know the formula 2 to the power of n. For suppose one bacteria 20 minutes are required. Right? Every 20 minutes the number of bacteria cells are doubled. Initially 1, next one 2, next one 4, next one 8, next one 16, next one 32, right? Next one 64, like this. So continuously the number of cells are doubled by the binary fission. Right? 1 lakh becomes 2 lakhs, 2 lakhs becomes 4 lakhs, 4 lakhs becomes 6 lakhs. So immediately the cells are doubled by the binary fission. Right? Then presence of the gonal-less organism, naturally these have the red color. Okay, the organism have the red color, and these are also having that bioluminescence property. So when these are rapidly divided within the ocean region, okay, the sea surface exhibit the red color, whatever the sea, okay, and the tides are there, okay, the total region exhibit the red color. That is about that red tides in Mediterranean Sea. And during cell division, these organisms releases toxins. See, suppression of other species. That is the concept here. When these are growing rapidly, that the nutrients are required. Is there any other organisms also growing surrounding? Then maybe there is a nutrients deficiency will take place that, okay, in that region. So these organisms are releasing some of the toxic compounds. The toxic compound is the saxitoxin. That saxitoxin kills fishes and marine organisms. The saxitoxin kills fishes and marine organisms. Right? So that is about the saxitoxin. So how does saxitoxin have the capacity to kill other organisms? See, which are especially act on nervous system of the other organisms. So this is one of the neurotoxin. See, I like to discuss about different types of toxins are there. For suppose we know about the snakes. There is a crate. Okay, scientific name is Bangladesh. In Telugu, this one is called Katlapon. Right. See, that snake produces one kind of toxin. That toxin acts on nervous system. Create the toxin, act on nervous system. Viper, we know about the viper, it's having the toxin. The toxin contains protein degrading enzymes. Once the toxin is entering into our body, whatever the blood cells are there, tissues are there, all these are destructed, because the toxic compound contains some of the protein degrading enzymes. The toxin contains the protein degrading enzymes and degrades total blood tissues and remaining other tissues also. Right? And we know the king cobra, that toxin also act on respiratory tract, majorly act on the respiratory tract, and also act on nervous system. So here concept is different types of toxins are there and which are act on different systems. So saxitoxin produced by the dinoflagellates, especially gonal-less organism. Right? This is a neurotoxin, kills the fishes and other marine organisms. So that is about the dinoflagellates and the